carbohydrates structure and function. The course is mainly about metabolism. So metabolism of proteins, metabolism of carbohydrates, and the metabolism of lipids. But we need to lay some foundation, do some revisions of some things which you did in the first year before we go into the metabolism itself. So just like we did with proteins, we did starting with the structure and function. And then after that, we can go on into the metabolism of carbohydrates. In order not to make not to make it boring, so that we don't just list some functions of carbohydrates in general, and then that is it. What I try to do is that as we talk about the structure of carbohydrates, we pick some specific examples of carbohydrates. I mean, they're very simple ones, and talk a bit about their functions. I think that will be more interesting than just stating broadly functions of carbohydrates. Because particularly, you know that carbohydrates are a source of energy, and if it's only that energy which is in terms of metabolism of the carbohydrates, and then maybe you also want to talk about the fact that carbohydrates form components of the structure of cells. If you just say those broad things without looking at certain specifics, I think it will be boring. So we'll take some specific examples and then we look at their functions. Of course, it's just a period of about 2 hours, and even 2 hours. I can't be talking continuously for 2 hours. Maybe I talk for at most 1 hour 30 minutes. The rest of it I give to you. You speak, I listen to you, or you write in the chat, I respond to you. And so there's a lot to say about carbohydrates that we can't say that over just this short period of time we'll finish with everything about functions of carbohydrates. But we'll do our best. We'll take a look at some very basic things we'll look at, and also so that we can relate it to the next part of this, which is the metabolism itself. So it will be more interesting that way. Now we start to look at the structure of carbohydrates, starting from the basic units of carbohydrates being monosaccharides. The same way as you refer to the basic units of proteins as amino acids, the same way you have the basic units of carbohydrates being monosaccharides. So it is these monosaccharides which are the monomers, which will get polymerized. So monosaccharides will bind together to form a large molecule of a carbohydrate. And the word monosaccharide is coming from Greek, monos meaning single, and then saccharine meaning sugar. So to say single sugar, that's what it means. Now, you realize that the simple sugars that you come across in their naming, you have the suffix "ose" O-S-E at their ends. So for instance, the commonest sugar you talk about is glucose. It ends with O-S-E. See another one, fructose, which is in fruits, galactose, etc. So if you look at the names of monosaccharides and disaccharides, even disaccharides, because common sugar is sucrose. It ends without OSE. If you take lactose, lactose will be a combination of glucose and galactose. So you realize that all these, even two glucose molecules binding together, give you maltose. So when you see the name of any of these compounds, looking at the suffix, you can tell that that particular compound you are talking about is a carbohydrate. It's a simple sugar. Now, the chemical formula of monosaccharides it can be looked at as a conjugation of carbon and water molecule. And you have X representing number of carbons, such that the number of carbons will be the same, will be equal to the number of water molecules that are inside the monosaccharide molecule. I mean, this in the first year, that the carbohydrates are polyhydroxy molecules. What it means is that you have a lot of hydroxyl groups, so apart from hydrogen. So you see, if you look at this chemical formula, what you can simply say is that the monosaccharides and in general, carbohydrates consist of carbon, hydrogen, and oxygen. And this general formula helps you to see the proportion in which the elements, carbon, hydrogen, and oxygen are. And you can see some simple monosaccharides like glyceraldehyde. Glyceraldehyde is one of these intermediates you find in the metabolism of carbohydrates. Itself per se, glyceraldehyde itself per se, it's not you can't say it's a carbohydrate, but when we refer to it as a triose, it's like a carbohydrate consisting of 3 carbons. And that's because it's in that pathway of carbohydrates. When you're looking at a breakdown of carbohydrates, you will see glyceraldehyde along the line. And you can see clearly that 3 carbons. So if you take this general formula, you see the number of carbons here is 3. And remember, this hydrogen is 2. So 2 times the X, that's the number of carbons, will give you 6. And the number of carbons is the same as the number of oxygens that you have. So oxygen there is also 3. And you see that with all. So if you have a 5 carbon sugar like ribose, what you have in nucleic acids, RNA. So in the nucleotides, you see this ribose. You see carbon 5, and the hydrogen is twice the number of carbons. Oxygen, same number as the carbon. And it runs through, even with the 6 carbon sugars, glucose, fructose, galactose, these monosaccharides are isomers. But you see that they have the same chemical formula because they all contain 6 carbons. It's just that their structures are different. So as we continue, we can monosaccharides in terms of the number of carbons that are present in their molecule. So if you take monosaccharide. So like what we were looking at, glyceraldehyde, that I said it's an intermediate in the metabolic pathway of carbohydrates, where you have X being number of carbons. When you have that being 3, number of carbons being 3, you refer to those carbohydrates as trioses. So apart from glyceraldehyde, you also see another triose, dihydroxyacetone, which is also in the pathway of the breakdown of carbohydrates. You have tetroses, which consists of 4 carbons, pentoses, 5 carbons, like those pentoses you find in the nucleotides. Hexoses, fructose, all of them, they consist of 6 carbons. Higher than that, like heptoses, this usually is, you don't find a lot of sugars that are heptoses, except that, well, in some bacteria, you have some heptoses being produced over this, are not sugars that will be existing much in like naturally, except that you have no, same thing as optosis. Also, you see that those sugars really are not purely monosaccharides, like carbohydrates in nature, but except that they are derivatives. They are bound to something else. They have some they have formed some conjugation and acting as some structural element or some carrying some functional property within maybe cell membrane of some the single cells like bacteria, things like that. You'll find some of this. In fact, the most commonest that we talk about are hexoses. And then pentoses, you talk only about 2 of them that are found in nucleic acids. And then trioses, which are those 2 that we mentioned, like the glyceraldehyde and the dihydroxyacetone. The rest of them, they are not common per se that we talk about. If maybe you are referring to some medicine, some drugs, which are of bacterial source, for instance, those ones, you see that they'll be having structures monosaccharides, which are in bacteria, like produced by certain groups of bacteria as a form of protection, against some host or some other, bacterial or viral attack. Right. Now, you also want to remember, I know you did this in first year, the classification of monosaccharides based on the functional group which is present in the monosaccharides, where you have monosaccharides ending, having a monosaccharides with a ketone group being ketoses. So you have that kind of classification as well, aldoses and ketoses. So for instance, if you take the simplest of these carbohydrates that we are referring to, the trioses, those containing 3 carbons, you see that glyceraldehyde is having that terminal group, an aldehyde group, carbon, double bond, O, and a hydrogen on it. That's an aldehyde group there. And we refer to that as an aldotriose. It's a triose because it contains 3 carbons, and it's an aldose because it's having the aldehyde functional group. So it's an aldotriose. If you take dihydroxyacetone, it's a ketotriose. It's a triose because it contains 3 carbons, and a functional group there is that carbon, along the chain, having a double bond on it. That's a ketone group. And the interesting thing which you should keep in mind, when you're referring to an aldehyde and a ketone, the aldehyde group will always be at the end, to be at the terminal end of a molecule. A ketone group cannot be at the end of a molecule because a carbon that carries the double bond O has to be bonded to, along the chain, but not at the end, at the terminal end of a molecule. Now we can also remember, remind ourselves of what we refer to as enantiomers. So enantiomers, these are stereoisomers that are mirror images of each other. So their structures are, non-superimposable. So you see that you have what we are seeing here is just what we refer to as ball and stick structures. Of course, you can do this with a simple molecule like this just for illustration, just to show. But of course, this is not how we write, chemical formulas. This will take you a lot of time to be doing. So we have what we refer to as Fischer projection formulas, named after a German chemist, Emil Fischer, who developed this form of representation of molecules just by, using the dash lines showing the bonds between the carbons and the other groups that are on the carbons. That's a Nobel laureate there. There are so many things as a chemist he did that if you check on Emil Fischer, you see a lot of things that he did in chemistry, in biochemistry, that up to today, you see his name being named after so many things. Excuse me. With a Fischer projection such that, the central carbon there, what you refer to as a chiral carbon, because it's having 4 different groups on it. And you realize that apart from the horizontal groups, if you look at the groups vertically, they are the same in them. So the CHO is referring to the aldehyde group. That's a glyceraldehyde molecule, the CH group, CHO, and then you have the CH2OH there. Now what you see is that, this molecule, D-glyceraldehyde, is a mirror image of L-glyceraldehyde. If you draw a mirror line, between the 2 molecules, then you see clearly that these are mirror images. So these are enantiomers being shown in Fischer projections. If you want to remember this, the only thing is, the D-glyceraldehyde is having the hydroxyl group to your right-hand side, and the L-glyceraldehyde is having the hydroxyl group to your left-hand side. So that's how you can remember this thing. And in fact, when you take other molecules, even like proteins, for instance, or any large molecule, and you want to represent the D and L isomers, this is used as a standard, such that you only have to, look for the chiral center, and then you place all the groups that are around it, that are on it, using this same standard, say you have the large groups, pointing in horizontally, and you have, if there's some hydrogen or hydroxyl group, you show them, hydroxyl group pointing to the right-hand side. That will be the D isomer. And then the L isomer will have the hydroxyl group pointing to the left-hand side. Or another way is to arrange the group such that you read the groups clockwise, depending on how large they are. You arrange it in that manner, and then you can say that is a D isomer or an L isomer. Another way of representing these isomers is by what you refer to as the Natta projections, named after the Italian chemist Giulio Natta. Now, what you see here is these thick bonds, the bold ones on the horizontal, are bonds that are pointing towards us. So this is a form of representation of molecules in a 2D structure, to see that you have the bonds that are the dash wedges, showing that those groups that are on those bonds are pointing away from you. Like as you are looking at your screen, like you see the molecule is not it's not flat. It's not just something which is on a paper. A molecule is a real thing which is having like a 3D structure. But if you are using a paper to represent it, you can look at it as a carbon is on the plane of the paper, and the group the aldehyde group is pointing away from you. CH2OH group is pointing away from you. Those dash wedges show that the bonds are pointing away from you. And the thick bonds, those bold ones, are pointing towards you. So like you can see it more clearly because it's pointing towards you. That's why it's bold. And the ones pointing back, because you cannot see that clearly, is getting far away from you. So it's shown with those dash lines. You can see here too, the groups are arranged the same way, except that the bonds are represented with these dash wedges and of somebody say you cannot hear my voice. Is it everybody who cannot hear my voice, or it's only one person? Well, you have epimers. Epimers are also some forms of their isomers that their structure varies only on one carbon when you take a look at it. So, for instance, mannose and glucose are epimers, in the sense that apart from the carbon number 2, if you look at the in mannose on carbon number 2 is pointing to the left, and in glucose the hydroxyl group is pointing to the right. That's the only difference between mannose and glucose. All other things are the same. Like you see, the hydroxyl group on carbon number 3 is pointing to the left in mannose. It's the same for glucose. And when you come to carbon number 4 and 5, the hydroxyl groups are pointing to the right. And it is the same that you are seeing also in glucose. So it's only on one carbon that you are having that difference. The same way if we take glucose and galactose. Glucose and galactose are also epimers. The only difference between them is on carbon number 4, where you have hydroxyl group in glucose pointing to the right, and the hydroxyl group on carbon number 4 in galactose is pointing to the left. All the others are the same. On carbon number 2, hydroxyl group is pointing to the right. It's the same for galactose. If you look at to the right, right, then in galactose, it's also pointing to the right. So the difference is on only carbon number 4. So when you have two monosaccharides that have difference of the orientation of the groups on one carbon, only one of the carbons being different, being opposite of each other. That's what you refer to as epimers. So when you say epimers, you cannot can be epimers, or one is an epimer of the other. That's that you can't you like, for instance, we are having 3 molecules here on the screen. You can't say all of these molecules are epimers. No, you can't say that. What you can say is that mannose and glucose are epimers. Then glucose and galactose are epimers. So epimers refer to 2 molecules that are having a difference in the orientation of the groups that are on them. Right. Now, apart from these isomers that we've talked about, if you are looking at all these structures of aldoses, starting from the 3 carbon, only one, triose there as an aldose. So of course, it's not having any isomer there. Okay, I saw that my screen went off a bit. I was monitoring it. I realized the presentation went off. That's why I kept quiet. So if you carbons, they are not mirror images of each other, but they are isomers. So this is what we refer to as diastereomers. So you can say the D-erythrose and the D-triose. The D-triose, the 2 of them, diastereomers. They are not mirror images of each other, as we saw starting from the beginning. Mirror images of each other are enantiomers. These are not enantiomers, but they are isomers, and we refer to them as diastereomers. So these are like the D isomers of the 4 carbon aldoses. Same is none of them is a mirror image of the other. The same thing if you take all of these 6 carbon sugars, you look at them, none of them is like mirror image per se. None of them is a mirror image. They are all different in their structures. But this we are looking at only the D isomers, which means that actually you can have L isomers. You see, these will have L isomers. So for instance, if you take, say, allose, see, the allose will have an L isomer. That L isomer will be its mirror image. So that will be its enantiomer, the L isomer of the allose. So if you take all these altrose, glucose, so you see you have D glucose, you can have L glucose, or you have D mannose, you have L mannose. That will become its enantiomer. But they themselves, like this, all the D isomers, we refer to them as diastereomers. They are not mirror images. They are not enantiomers of each other. Okay. Where again? Oh, patience. All right. You said you couldn't see the screen. I think that happened earlier, but now the screen is showing. So, okay. So actually, you see these structures that we're just looking at over here. You can actually draw all of this if you understand the system by which they do. So this is what we refer to as a family tree of aldoses. I hope it did this. Glyceraldehyde. That is D-glyceraldehyde. See, the hydroxyl group is pointing to the right. Now, all you need to do is that, you see, to come to the next stage means that you add one carbon to get a tetrose. Because the first one there, take that as first generation glyceraldehyde. And it's having that one para carbon in the middle. So, and then the hydroxyl group is pointing to the right. Now, isomers. In what sense? So if you add one carbon, and that carbon you are going to add will come right after the aldehyde group. You maintain all these other things here before are maintained except the carbon that you are coming to add. So for instance, this hydrogen with the hydroxyl group, which is pointing to the right, is what is maintained below here. Is the same also here. What is numbered 2A and 2B, you see the hydroxyl groups are pointing to the right. That group, that carbon in the middle has been maintained. Now, the new carbon which you are adding to get a tetrose is coming right after the functional group, which is the aldehyde group. You can either have the hydroxyl group on that carbon pointing to the right, or you have the hydroxyl group pointing to the left. That is in 2B. So you have the possible tetroses after that triose. And if you continue that way, somebody says they still cannot see my screen. Are you sure about that? Perhaps you are having an issue there, because as I monitor, yes, my screen went off for some time. It was not showing. I waited and then it came back. So as at now, I can see that the screen is back. Right. So if you take like the 2A, the 2A will also give you two possible isomers. Now, if you maintain these two, the two carbons in the middle there, those two carbons, those two carbons are maintained so that you have the hydroxyl groups which were pointing to the right maintained below here, hydroxyl group, hydroxyl group. The new one that you add is having a hydroxyl group on it pointing to the right. 3B will have that hydroxyl group pointing to the left. Same thing you do for 2B. You maintain this hydroxyl group pointing to the right and this hydroxyl group pointing to the left. That's what you see below. Hydroxyl group pointing to the left here and hydroxyl group pointing to the right. Then the new one that you are adding at the top is having hydroxyl group pointing to the right. 3D will be having the hydroxyl group on it pointing to the left. When you continue to do that, all of these, so if you say 3A will give you two possible isomers. All these hydroxyl groups pointing to the right in 3A, maintained below. The new carbon which is added, you either have the hydroxyl group pointing to the right, or in 4B, will give you two possible isomers. And you see there are two hydroxyl groups pointing to the right, and then one hydroxyl group at the top pointing to the left. Those ones are maintained below. Two hydroxyl groups pointing to the right, and one hydroxyl group pointing to the left. The new one that comes to add at the top, that carbon, is having hydroxyl group pointing to the right in 4C. In 4D, you have that group, which is in 3B, maintained, all those groups, hydroxyl groups pointing to the right below, and a hydroxyl group pointing to the left. The new carbon that comes to add at the top is also pointing to the left. All right, so I don't know what is happening. Some people are right. Somebody writes that you can't see screen. Another person will be writing, I cannot hear. Anyway, somebody is advising you that you should rejoin. Maybe you have some issue there. That's why. You take 3C. You take 3C. You see, same thing is happening. If you maintain this, what you have already in 3C, hydroxyl group pointing to the right, hydroxyl group pointing to the left, hydroxyl group pointing to the right. So these three, you maintain them below, hydroxyl group pointing to the right, hydroxyl group pointing to the left, and then another hydroxyl group pointing to the right. The new carbon, which you add at the top, is having a hydroxyl group pointing to the right for 4E. And in 4F, that new carbon you are adding, the hydroxyl group is pointing to the left. You do the same thing for 3D. You maintain the three chiral carbons, the orientation of the hydroxyl groups. See, hydroxyl group is pointing to the right below, and the two hydroxyl groups at the top there pointing to the left. When you come to 4G, that in 3D is maintained. Hydroxyl group pointing to the right, two hydroxyl groups pointing to the left, and a new one that you add, a new carbon you add, is having a hydroxyl group pointing to the right. That's in 4G. In 4H, that new carbon you add, the hydroxyl group is pointing to the left. So if you do that kind of thing, you can actually be able to draw the structures, of the aldoses. This is something which you should just sit down and be playing with and get to understand, that systematic structure, the structures of these aldoses. This is much of an importance for you to understand than just going to be cramping the names of these aldoses in your head. You should be able to, play around these and get it. It's easy. If you pay attention and you just maintain groups, and then the new carbon you add, you either put hydroxyl group to the right or hydroxyl group to the left to have the new isomers coming from each of those groups. So you have this from first generation coming to the fourth generation, which is the 6-carbon sugars. Same thing you can do for diastereomers of ketosis. The only thing is that for ketosis, the dihydroxyacetone is having no chiral center in it because there is no single carbon in dihydroxyacetone that has 4 different groups around it. For instance, if you take that central carbon, which is having a ketone, having a ketone group there, you only have 2 other groups on it. So those are 3 groups. It's not up to 4. If you take the first carbon at the top, CH2OH, already you can see there are 2 hydrogens, which will be the same. They are not different. So it can be a chiral center. The same thing for the terminal carbon, which is below. But when you add a new carbon, see that carbon again, you are adding it below the functional group. The functional group is a ketone group. When you add it, see the hydroxyl group is pointing to the right because it's a D isomer, D-erythrulose. If you go to 5-carbon sugars, see you are adding a new carbon right after the functional group, the ketone group. And you can see you maintain that carbon in erythrulose, you maintain it below there. So the orientation of the hydroxyl group to the right for erythrulose is the same that you see on that carbon on ribulose. And you see the D isomer here, you have the hydroxyl group pointing to the right. Now you see again D-xylulose is the same thing that we did previously that we are doing here again. You see that after you maintain that hydroxyl group on the right from erythrulose, the new carbon which comes after the functional ketone group, the hydroxyl group is pointing to the left. So that's for the 5-carbon sugars. You take the 6-carbon sugars, and for each of them, for D-ribulose, you do the same thing. For D-xylulose, you do the same thing. If you take ribulose, these two hydroxyl groups are pointing to the right. You maintain them. So in D cycles, the 2 hydroxyl groups below are maintained to the right. The new carbon, which comes right after the ketone group, the hydroxyl group, is pointing to the right. And for D-fructose, those 2 carbons are maintained, their configuration is maintained so that you have the hydroxyl groups pointing to the right, but the new carbon which you are adding, the hydroxyl group points to the left. So that's coming from the D-ribulose. Same thing for xylulose. You have that last chiral carbon, the one below there, is having hydroxyl group pointing to the right, and the second chiral group, which is right after the ketone group, you have the hydroxyl group pointing to the left. So you maintain those 2 chiral center configurations. Hydroxyl group pointing to the right below here, and then you have a hydroxyl group pointing to the left. The new carbon, which comes right after the ketone group, hydroxyl group is pointing to the right on it. That's on sorbose. That's in sorbose. But in tagatose, you see the hydroxyl group is pointing to the left. The configuration for those two carbons below is maintained from xylulose. So with this, you can have that family tree as well over there. Right. Now you also want to remind yourself of what is referred to as the Haworth structures. That's the cyclic structures. We have so far we've been looking at these monosaccharides in straight chain structures. Those are the Fischer, projections. But you have also the Haworth structures, or Haworth perspective formula, sometimes being referred like that. But that too, was developed by an English chemist, a British chemist, Walter Haworth. So from that, the Fischer projections, you can have cyclic structures being formed. Actually, these sugars exist as cyclic structures in solution. And you see, we were referring to those carbons in the straight chain structures as chiral centers, that you can have hydroxyl groups pointing to the left or right. Hydrogen and the hydroxyl group able to move around, like it can change its configuration. Either the hydroxyl group is pointing downwards or is pointing upwards, to have those two configurations. Where you have the hydroxyl group pointing downwards, you have an alpha configuration. So you see here the name is alpha D-glucopyranose. It's glucose and it's a pyranose. I think you also there, that's a pyranose. If you have a 5-sided ring with an oxygen, at the apex at the top, you have a furanose. I believe you did that in first year. And here you have a beta configuration. Glucopyranose is having the hydroxyl group pointing upwards. And that is possible because of what we refer to as mutarotation. There can be a rotation of this hydrogen and hydroxyl group on carbon number 1, which is also referred to as the anomeric carbon. And in that case, the alpha and the beta configurations are referred to as anomers. So we saw enantiomers, we saw epimers, we saw diastereomers. Now we are seeing anomers. So anomers are with the ring structures. Now, we've been talking about these monosaccharides themselves with these various stereoisomers of them. You have also what you refer to as derivatives of monosaccharides, where a hydroxyl group, as you can see, these monosaccharides are polyhydroxy. As I mentioned earlier, they contain a lot of hydroxyl groups. But when you have any of these hydroxyl groups in the structure of the monosaccharides replaced with something else, it becomes a derivative. It becomes so it's either you say a modification of the monosaccharide or a derivative of the monosaccharide. Now, it's important you get this because as we come to the metabolism of carbohydrates, you'll be seeing a lot of these derivatives. So the first one you are seeing over here is glucose 6-phosphate, which you will see in the process of metabolism of glucose. That's a phosphorylated glucose molecule. So see the red group there, the phosphate group, is on carbon number 6. That's why it is glucose 6-phosphate. You find it along the major metabolic pathway of glucose. The process will give you this molecule glucose 6-phosphate, which will then go down the reaction pathway. It's a form of activation of the molecule to go down a particular reaction pathway. So that is one of the derivatives can talk of. So now what you see the things we are talking about now, like we are carbohydrates, but you see these carbohydrates, they are not just standalones and acting, but you find these carbohydrates in complex with other molecules or other chemical entities and carrying out some specific functions. So in order to broadly speak about the function of these monosaccharide molecules or carbohydrate molecules, we also have to consider the complexes that they form with other biomolecules, like with proteins, where you have the formation of glycoproteins, or carbohydrates can bind with lipids and form glycolipids. You also see carbohydrates can bind with even in a biological system, you have all these biomolecules in complex. So they will be acting together in some complex. We see another derivative which we see on carbon number 2 for glucose. There's a hydroxyl group there. In place of that hydroxyl group, you now have an amine group. And that derivative is glucosamine, which will serve as a precursor for the synthesis of glycoproteins. That is a complex of carbohydrate and protein. And glucosamine is involved in the synthesis of the lubrication in the joints. And so perhaps some of you, maybe you have done some attachment in some clinical setting, some pharmacy, you may have come across glucosamine, which is prescribed to a lot of times people of age when you're having joint problems. They prescribe this glucosamine to them. So that's it's a pharmaceutical agent there you are seeing. And because of this amine group that comes to bind on the carbon number 2, it means that some peptides can easily bind onto this amine group. Sort of, the carboxylic acid group can come and bind here as if it's forming a peptide bond. And that will result into, the synthesis of glycoproteins. Another is N-acetylglucosamine. So that is just coming from the glucosamine. So from glucosamine, so you see what I was talking about? From glucosamine, you have an acetic group. So it's an acetic acid. See? So this is looking much like a peptide bond. And you have N pods, like insects, millipedes, centipedes. If you take the structure of their exoskeleton, you have an acetylglucosamine. as a major component of that structure. N-acetylglucosamine is also a major component in bacterial cell walls, forming what we refer to as peptidoglycans, particularly in the gram-positive bacteria. You will see that. All right. Now we'll see glucose. Another glucose derivative is muramic acid and N-acetylmuramic acid. So see, this is what I was even referring to. Like, see, here you have N-acetylmuramic acid and muramic acid forming what the peptidoglycan, which is a major component of a bacterial cell wall. And it's actually, being synthesized from N-acetyl. Glycan is that you have N-acetylglucosamine, which we saw earlier, and N-acetylmuramic acid alternating and giving you that glycan chain. And then between these glycan chains, you have, some peptide linkage to give you, a certain cell wall structure, giving protection to bacteria. So we are looking at the functions of carbohydrates and at the same time looking at derivatives, their structures. And what do you notice? You notice it's the same as what we saw with N-acetylglucosamine, what we saw earlier. Then something else. See, there's an R on carbon number 3. That R is a residue of lactic acid. So lactic acid is also represented here for you to see. And muramic acid is rather, glucosamine, because on carbon number 2, you have the amine group is glucosamine with that lactic acid residue on carbon number 3. So that's the difference between N-acetylmuramic acid and muramic acid. See, the N-acetylmuramic acid is having an acetic group on that carbon number 2, binding on that amine group. But you have in muramic acid just the amine group with the lactic acid residue. Another one is glucuronate. It's an acidic sugar. And what do you see? What you see is that, see, the carbon number 6 of the glucose molecule is oxidized. We see in glucose molecule that carbon there, the group there was CH2OH. But here, you see it has become it has been oxidized to a carboxylic acid group in glucuronate. It's an acidic sugar. You find it in first isolated in urine, and it's involved in the process of glucuronidation. This is a conjugation reaction. It's a type of detoxification reaction that occurs mainly in the liver when you take in xenobiotics. That is when you take in compounds that are maybe of a medicinal value, like when you take in medicine, or even if it's not of a medicinal value, maybe it's toxic. And the liver is responsible for the metabolism of these foreign substances coming into the body. And one of the processes is a form of a conjugation, is for these glucuronic acid to bind onto the molecule, forming a metabolite, which then will get excreted. And mostly these excretions occur through the kidneys. So from there, is found in urine. So that's why it says first isolated in urine. Then you can also have gluconic acid or gluconate. Uh, the ring structure opens. It's not a ring structure per se. And then you have that first carbon, which actually originally, remember that the glucose molecule is an aldose. So this group originally is an aldehyde group, which means that this group as an aldehyde group gets oxidized to a carboxylic acid group to give you that gluconate. And that you find naturally occurring in fruits, in honey, in wine. And sometimes it's used in the food industry as an acidulant, or sort of regulating acidity, preventing the growth of bacteria in foods. It's also used in the production of an injection, what we refer to as the zinc gluconate. In neutering of male dogs. I mean, neutering of male dogs only refers to chemical castration of the dogs. All right, so that is gluconate. Now you can have also gluconolactone as another derivative, lactone. So lactones will have a cyclic ketone group. So you have the ring structure, all right, and then on that anomeric carbon, where you were having one hydrogen, one hydroxy group, you now have a double bond O, a ketone group. So still some oxidation has occurred there. And this is also used in the food industry as a sequestrant. A sequestrant is, these are substances that are used for the binding of metal ions, like chelating agents, like EDTA, so that they prevent oxidation in particularly oily foods that contain oil, so that you don't have oxidation of these and even texture, losing this also, just like what we saw earlier, that you can have what we refer to an acidifier or acidulant, sort of controlling pH, leavening, like in the bakery. You see when they are doing the cakes, you have some of these leavening they use, which sort of causes the cakes or the pastries they do to be fluffy. That is some leavening there. Curing, just addition of these lactones, sort of preservation. Just like how you add salt or sugar, to foods to preserve them, or pickling. In Ghana, we don't do pickling. They say that like you take some fruits or vegetables and you store it in a jar with brine, like salt water, for instance. Close it tightly away from oxygen, keep it. You can then eat it later. Let's say usually you find this being done in temperate regions, so that in summer when it is warm, they pickle the fruits and the vegetables, and then when it is winter, vegetables and fruits. Now you have also what we refer to as, so well, we already saw some amino sugars because we saw glucosamine. Now we are looking at galactosamine. Just like glucose with the amine group, you have the galactose with the amine group on carbon number 2. If you remember, you saw glucose and galactose as epimers, and it was on carbon number 4 that galactose and glucose were epimers. So you see here on carbon number 4, where you have the hydroxyl group pointing upwards in galactose, in glucose, that hydroxyl group will be pointing downwards. That's the only difference between them. And these derivative, galactosamine, is found, or let's say it's a precursor for the synthesis of hormones. There are some hormones which are glycoproteins. Then it is, galactose that forms conjugation with some proteins. Examples of such hormones are the follic stimulating hormone, luteinizing hormone, playing some roles in secondary sexual characteristics. But you have galactosamine also having some hepatotoxicity, so that in the lab, a lot of times when things like hepatitis used is injected as an inducer of hepatotoxic effects. So practically, galactosamine is used in the lab for such purpose. Manosamine. The manosamine itself does not have some specific function, except that it serves as a precursor for acetylmanosamine. So we can just move quickly to acetylmanosamine. So acetylmanosamine actually will go into, say, the synthesis of N-acetylnuraminic acid. Look at the name. In the name, you can even quickly tell where you can find it and what is possibly to be doing. Say N-acetylnuraminic acid. When it's nuraminic, so in neurons, nerve cells. So it's a component of gangliosides. These are some nerve cells in the central nervous system. And you see here you have a N-acetyl group on carbon number 4. There's some R group, which is on the group is represented there. 3 carbons there, like a triose. Looks like a residue of a triose there. That's coming on carbon number 5. Then there's a carboxylic acid group, which comes on this carbon number 1. There's a carboxylic group which comes on it. So that is an acetylnuraminic acid, which is a component of granulocytes. Like you find this in the cell membrane of some nerve cells responsible for the transmission of nerve signals in the central nervous system. Fucose. So these two, you can see, what you see is that you see that CH2OH, which was the carbon number 6, it has gotten reduced to CH3. See that? CH3. And it has even changed its configuration. It's now pointing downwards. In glucose, you have this as CH2OH pointing upwards. Yeah. Fucose, which is found in seaweed, fucose. So first extracted from this plant. That is why it's been given that name, fucose. This you'll find as a component of antigens. So actually what happens is you have structures of antigens, like you see when you take blood, like the blood groupings, blood AB, the ABO blood group. If you go and look at the structure of the antigens that are responsible for this blood grouping, you see that those structures, those antigens have some sugars that are bound together, giving you some oligo saccharides, forming some complex of I think some glycolipids there, some glycoproteins. And you have fucose, which sort of is at the terminal end of the structure, which actually, when it is at the terminal end, it's responsible for cell recognition, to recognize like some foreign material, like infection, and then begin to fight it. So it plays a major role in the immune system. So in fact, there is even some engineering of this, such that you have fucose removed from some of these antigens introduced into the body, so that for cancer cells, for instance, when it goes to reduce the cell recognition in these cancer cells, it means that those cancer cells will lose their immunity. So it's sort of inducing in vivo cell death. So because then you have antibodies that can quickly attack these cancer cells. So that is a practical application of I've been talking, I think. So I have to just end somewhere, because there's no way we can finish all of this today. We can quickly take a look at, say, maybe deoxy sugars. So that's another one, rhamnose. I mean, you may want to see, so what's the difference between this and this? The hydroxyl group here on carbon number 1 is pointing downwards. Here is pointing upwards. So that's already one difference. Maybe if you look further again, these two hydroxyl groups, carbon number 2, carbon number 3, are pointing downwards. It's not the same here. You have this hydroxyl group pointing upwards, or fucose. in which the hydroxyl groups are facing, you see the differences in these structures. Also isolated first from a plant, Rhamnus species. So that's where the name comes from. Still is also present in some microalgae. It's a component of the cell membrane. So if you take this single cell organisms like Mycobacterium species, you'll find Rhamnus in their cell membranes, as a component of the structure of the cell membrane. You find that. So that's a function that you can talk about. Fructose 1,6 biphosphate. These two, you come to see it when we go into the metabolism of carbohydrates. We see the fructose 1,6 biphosphate as an intermediate in the process molecules and further to go and give you the last product, pyruvate. We'll come to it, so you'll see it. So now at least you are seeing some names, you are seeing some structures, telling you that this exist, and you already will know how they look like in a way. So it's a foundation we are building before we come to look at the metabolism itself. So yes, this is one of the things you will be seeing in the process of glycolysis. That will be in the catabolism of carbohydrates. Wow, I think that we should leave this so that when we meet next time, we continue from here. Because from here, we go ahead and then look at, okay, all the way up to, say, formation of polysaccharides. So the next time we meet, we'll start from here, disaccharides. So I'll end here for now so that I keep quiet and listen to you. So far, we have been listening to only me for more than 1 hour, close to 1.5 hours. So I'll keep quiet now. The floor is open. If you have any questions, you have any contributions, you have any issue, anything you want to say, you can write in the chat, or you can raise your hand. I will enable your microphone, call your name, and you speak. The floor is open.